Good day, sir. I'm Cursinger Flora Malano from BS Criminology 2 Bravo. So, I'm here to discuss about the microscope. What is microscope? Microscope are instruments that are used in science laboratories to visualize very minute objects such as cells, microorganisms, giving a contrasting image that is magnified. Microscopes are made up of lenses for magnification, each with its own magnification powers. Depending on the type of lens, it will magnify the specimen according to its focal strength. So, yung microscope is ginagamit ito para makita yung mga maliliit na cells, maliliit na microorganism na hindi nakikita na visualize ng ating mga mata. So, yung gamit ng microscope is titignan, sabi ko nga, titignan yung mga maliliit na organism, specimen, ganon, yung mga cells, yung mga ganon. So, microscope, their ability to function is because they have been constructed with special components that enable them to achieve high magnification levels. They can view small specimens and distinguish their structures difference. For example, the view of an animal and plant cells, viewing of microorganic bacteria cell. So, gaya nga ng sinabi ko kanina, yung function, the other function of microscope is to titignan yung mga maliliit na cell, gaya ng mga plant cells, yung mga animal cells, ganon. Para makita, hindi na visualize ng ating mga mata, para makita nila yung mga maliliit na cells, ganon. So, the other meaning of microscope are generally made up of structured plants for holding and supporting the microscope and its component and the optical part, which are used for magnification and viewing of the specimen images. This description defines the part of a microscope and the function they form to enable the visualization of a specimen. So, gaya nga ng sinabi ko kanina, yung microscope is ginagamit para makita yung mga cells, microorganism na hindi nakikita na visualize hindi nakikita ng maayos ng ating mata. Hindi, what I mean is, for example, di ba ang ating mata is na visualize, nakikita mo isang ibagay, like for plant, sana, di ba, nakikita mo siya plant, pero yung plant na yon may cell pa, makikita mo yung mga cells niya, yung parang ganun. Yun yung gamit ng microscope. The other example is yung urine. Urine, di ba? Titignan mo, another example na lang is yung sperm. Nakikita mo kung may bacteria or kung ano man yun, parang makikita mo yung mga bakterya na maliliit na hindi nakikita mismo ng ating mga mata. But I mean, hindi mismo, andyan sa harap mo, is hindi ganon. So, yung microscope is yun nga, para makita yung bakterya na hindi nakikita ng ating mga mata. So, next is the parts and function of a microscope. So, the first part of the microscope is the body tube. It's a hollow tube through which light travels from the object to the ocular. It contains a prism at the base of the tube that bends the light rays so they can enter the inclined tube. So, yung body tube, like for examples. This is the body tube. Yan. Ito yung body tube. So, yung body tube is siya yung inclined. Kasama niya yung the eyepiece. Dito din yung ito yung parts of a microscope. So, dito sa body tube, siya yung ginagamit, siya yung pinaka main paghahawakan mo, siya yung kasama niya yung eyepiece. So, yung next naman na part is the nose piece. This circular structure is where the different object lenses are screwed in to change the magnification power, simplify, rotate the current. So, dito, sa aking ginuhit, ito yung the nose piece, revolving nose piece. So, yung gamit ng revolving nose piece, siya yung siya kasama nung the objective. Siya yung taga-handle ng objectives. Siya yung taga-hold. Siya yung nasa itaas ng objectives. Siya yung i-rotate mo siya if makikita mo ba sa malayo, sa malapitan, yung gusto mo, ganun yung nose piece. So, the next is the low power objectives cover a wide view of view, and they are useful for examining large specimen or serving many smaller specimen. So, dito nga kanina yung sinasabi ko, ito yung revolving nose piece. So, dito sa revolving nose piece, ito yung dalawang objective niya, the low power objective and the high power objective. So, yung low power objective, siya yung makikita mo yung mga malapitan ba, yung mga large specimen, yung parang ganon. So, next naman is the medium power objective is attached to the nose piece and magnifies 4 times. So, dito nga sinasabi ko kanina, yung kasama ng revolving nose piece, siya yung revolving nose piece, kasama niya yung low and the high power objective. So, yung high power objective naman, dito mo makikita mo sa malapitan, pwede mo siyang parang na-zoom na yung parang ganon sa high power objective. So, the next is the stage clip. The flat platform that support the slides. Stage clips hold the slides in place. If your microscope has a mechanical stage, the slide is controlled by turning 2 knobs instead of having to move it manually. One knob moves the slides left and right. The other moves it forward and backward. So, yung stage clip. So, dito, stage clip. Nandito yung stage clip. So, yung gamit ng stage clip, siya yung taga-clip para makuha yung parang example nito is yung parang ganito, para siyang ruler, ilalagay mo dito yung cells, lalagay mo siya, then para ma-fix niya, yung parang clip na maliit, pwede mo din siyang i-move backward, forward, or to the right, to the left, yung ganon, para ma-support niya, para ma-clip niya, para ma-clip niya ng maayos, para hindi gagalaw-gagalaw yung clip na yon, yung tinitignan mong cells, yung ganon. So, yun yung stage clip. So, next naman is the diaphragm or iris. The diaphragm or iris is located under the stage and is an apparatus that can be adjusted to vary the intensity and size of the cone of lights that is protected through the slides, as there is no set rule on which setting to use for a particular power. The setting depends on the transparency of the specimen and the degree of the contrast you desire in your image. So, yung diaphragm, kikita natin, ito yung diaphragm sa may baba siya ng stage. So, yung diaphragm na yon is siya yung taga-kita, taga-support para makita mo yung microorganism na tinitignan mo. Yung ganun yung diaphragm, pwede mo din siyang, hindi mo siya pwedeng i-rotate, yung parang ganun. So, next naman is the mirror. Microscope light source located at the base, it is used instead of a mirror. So, yung mirror naman daw, dito yung mirror. Dito siya nakalukit. Ang use nun para ma-detect, para ma-reflect yung light, para makita mo mismo yung tinitignan mong organism na tinitignan mo para makita mo yung mga bacteria, yung mga cells na hindi nasa sight ng iyong mga mata, na hindi nga as in nakikita mo. Kasi yung mga other, for example, yung animal cells, hindi mo naman nakikita yung cells niya nun, di ba? If kundi gagamit ka pa ng microscope para makita mo yung cell, yung maliliit na cells ng animal cells na yon, di ba? Yung ganon. So, the eyepiece, also known as the ocular, this is a part used to look through the microscope. It's found at the top of the microscope. Its standard magnification is 10 times, with the optical eyepiece having magnification from 5 times to 30 times. So, yung eyepiece naman dito, eyepiece. Dito mo siya titignan, isa-sight mo na ganun yung organism, yung cell na yan, para makita mo siya. Pwedeng malapitan mo na tignan, or pwede mo siyang makita in malayuan, yung parang malayo talaga. Makikita mo yung nasa paligid ng organism na yon. So, that is the use of eyepiece. So, next is the arms. This is a part connected the base and to the head of the eyepiece tube to the base of the microscope. It gives support to the head of the microscope, and it is also used when carrying the microscope. So, yung arm daw, arm ng microscope, ito yung arm niya. So, yung use ng arm, dito mo siya hahawakan if bubuhatin mo yung microscope dito. Parang ganyan. Ganyan. Bubuhatin mo yung microscope. Dito din siya, hinahandle niya din yung body tube. Dito din yung spang support ng base. So, yung arms is, yun nga, hahawakan mo siya, hold. Para mahawakan mo yung, para mabuhat mo yung microscope. So, yung stage naman, this is the section of which the specimen is placed of viewing. They have stage clips that holds the specimen slides in place. The most common stage is a mechanical stage, which allows the control of the slides by moving the slides using the mechanical knobs on the stage instead of moving it manually. So, yung stage daw, stage. Ito yung stage. Stage. Ito yung stage. Yung gamit ng stage, siya yung taga-hold ng stage clip, at saka siya din yung taga-hold ng yung rack and pinions substance. So, yung stage daw, yun na, siya yung taga-hold ng stage clip, yung ganon. So, yun yung stage. Next is the curves adjustment knob, a rapid control which allows for quickly focusing by moving the object lens or stage up and down. It is used for initial focusing. So, yung curves focusing microscope, ito. Yung iba kasing microscope is hindi dito sa taas yung curves at focusing mechanism niya, kundi andito sila sa baba. Magkadikit itong curves focusing mechanism at saka itong fine focusing. So, yung curves focusing is, siya yung i-adjust mo, i-focus mo yung malaking, para yung mga maliliit, para i-focus mo siya, i-rotate mo siya, ganon. So, yung curves focusing mechanism yon. So, the next is the fine adjustment knob. This knob is inside the curves adjustment knob and used to bring the specimen into sharp focus under the low power and used for focusing when using high power lenses. So, dito naman sa fine adjustment, dito kasi fine adjustment. So, ito naman is i-focus mo siya if low lenses yung ginagamit mo. Kapag focusing mechanism, siya yung pinaka-high, yung medium power adjustment. Yung dito sa may objective, pag i-focus mo daw siya gamit ng fine focusing, yung fine adjustment is para gagamitin mo lang siya pag low power adjustment yung gagamitin mo. Makikita mo siya sa malayuan, yung ganon. So, yun yung ibig sabihin ng fine adjustment. So, the last naman is the base. Base, it acts as microscope support. It also carries microscopic illumination. So, ito yung base. Ito yung pinaka-base niya. Base ng microscope. Siya yung taga-handle, taga-hold ng arms ng microscope. Siya yung taga-support. Ito yung support niya. Support niya sa lahat ng parts of microscope. So, yun yung base ng microscope. So, the next is the different types of microscope. So, gaya nga nung kanina, gaya nitong example nito is, gaya din nung picture kanina sa microscope is may iba't iba silang klase. So, atin nang tatalakayin yung mga iba't ibang klase ng microscope. So first is a simple microscope. Is a magnifying glass that has a double convex lens with a short focal length. The example of this kind of instrument include the hand lens and the reading lens. When an object is kept near the lens, then its principal focus with an image is produced, which is erect and bigger than the original object. So, dito sa simple lens, dito daw is yung pwedeng sa reading, pwede din sa, ito yung pinaka, hindi naman sa una, pinaka-simple microscope. Pwede mapa-reading, pwedeng pagtitignan mo lang siya, yung parang ganon. So, yun yung simple microscope. So, the second is the biological microscope. Most of us remember using a microscope at some point in school, usually in biological class or a science class that include a section on biology. And for most of us, it was the same kind of microscope. A biological microscope is one kind of compound microscope. Sometimes, other names are useful for biological microscope too. If you hear either of the terms bright field microscope or transmitted light microscope, it referring to the same things. So, yun po yung biological microscope. So, the third is the stereo microscope. Is used for low magnification application allowing high quality 3D observation of objects that are normally visible to the naked eyes. In life science stereo microscope application, this could involve the observation of insect or plants. So, yung stereo microscope is dito inoobserbahan, ginagamit para sa mga insects, sa mga animals, at saka sa plants. So, ang stereo microscope is, siya yung para makita mo yung animal cells, plant cells, yung parang ganon. So, yun yung stereo microscope. So, the fourth is the conjugal microscope. Is widely used for fluorescence imaging in life science. The last decade has seen advances in illumination sources, detector, fluorescent probes, optics, and sample preparation techniques, which wide improvement in different combination of speed, depth, and resolution. So, yun yung fourth types of microscope, the conjugal microscope. The fifth is the scanning electron microscope, or the SEM, scans a focused electron beam over a surface to create an image. The electrons in the beam interact with the sample, producing various signals that can be used to obtain information about the surface topography and composition. The last one is the transmission electron microscope, or the TEM, or microscope that uses a particle beam of electrons to visualize a specimen and generate highly magnified image. TEMs can magnify objects up to 2 billion times in order to get a better idea of just how small, that is, think of how small a cell is. So, yun po yung TEM. What is the difference between TEM and SEM? So, TEM enables data to be captured regarding the sample's inner structure, where SEM provides data about the surface of the analyzed object. TEM offer a greater resolution compared to SEM. In comparison to SEM, TEM has more stringent requirements concerning the preparation of sample and condition of analysis. Next naman is the similarities between TEM and SEM. Both TEM and SEM shared several components. Multiple electrostatic and electromagnetic lenses are involved in magnifying the trajectory and shape of the electron beam. An electron surface, the sample chamber, is placed under the high vacuum electron captures. So that's all. Thank you for listening. I hope you understand. And keep safe and God bless.